Hello, friends. Today we're going to talk about RNA, ribonucleic acid, not to be confused with DNA, deoxyribonucleic acid. Let's get started. Genes contain coded DNA instruction that tells how to build proteins. Keep in mind, genes are those factors that get passed on from generation to generation that determine our traits. Like, do we have curly hair? Are we going to have certain color skin? Okay, so genes contain DNA instructions. The first step in decoding these genetic instructions is to copy part of the base sequence from DNA to RNA. So to get these instructions, we have to get them from DNA and we have to turn it into RNA. RNA, like DNA, is a nucleic acid that consists of long chains of nucleotides. Okay, so we talked about, remember our four macromolecules: carbohydrates, proteins, lipids, and nucleic acids. Well, that's our DNA and our RNA. And we talked about monomers, and the monomer for a nucleic acid is a nucleotide, which has a phosphate, a sugar, and a base. RNA then uses the base sequence copied from DNA to direct the production of proteins. So comparing DNA and RNA. Each nucleotide in both DNA and RNA is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base, which we talked about. There are 3 important differences between RNA and DNA. Kids, please pay attention. The sugar in RNA is ribose, not deoxyribose like in DNA. It's ribose, so the sugar is different. Number 2, RNA is single-stranded, not double-stranded like DNA. There's only one strand. And number 3, this is very important, RNA contains uracil in place of thymine. So we don't have apples to trees and cars and garages. We have apples under trees and cars and garages. These chemical differences make it easy for enzymes in the cell to tell DNA and RNA apart. The role played by DNA and RNA are similar to the plans of blueprints used by builders. So in other words, the DNA, it has the information, and then it copies the information and sends it outside the nucleus, okay? So DNA has the information. The DNA is very valuable, so it turns its information into a copy that they give to the RNA to go outside the cell's nucleus. You can think of RNA molecule as a disposable copy of a segment of DNA, a working copy of a single gene. RNA has many functions, but most RNA molecules are involved in creating proteins. RNAs control the assembly of amino acids into proteins. Remember we talked about macromolecules and monomers? Amino acids are the monomer of proteins. And remember what part of the cell, organelle of the cell, that makes proteins? Remember, it was the ribosomes. Each type of RNA molecule specializes in a different aspect of this job. There are 3 kinds of RNA, so pay attention to this. There's messenger RNA, ribosomal RNA, and transfer RNA. And I want you to notice here the amino acid at the top of transfer RNA. Now, this messenger RNA that you see here, you notice you see uracil. Instead of thymine, you see uracil. So this is messenger RNA, and it's made from the DNA. Then the messenger RNA leaves the nucleus and goes out to a ribosome. And the ribosome has a couple parts to it, okay? And then the transfer RNA, okay, hooks up to the messenger RNA. Now, how does that happen? We'll talk about that in a later video. But this messenger RNA has a code given to it by the DNA. In the meantime, the transfer RNA carrying the amino acid also has like an anticode which matches up with the code on the messenger RNA. Okay, so how does the cell make RNA? In transcription, segments of DNA serves as templates to produce complementary RNA molecules. Now remember that transcription. So we're going to transcribe DNA into RNA. Okay, there's going to be 2 terms you need to learn. Transcription, where the messenger RNA gets made inside the nucleus from the DNA, and translation, where the messenger RNA goes out to the ribosome and creates proteins. So keep those two terms in mind, transcription and translation. Transcription takes place inside the nucleus and makes RNA from DNA, and translation is where the RNA makes proteins. Here's something to keep in mind. DNA to RNA to protein. One more time. DNA to RNA to protein. That is very important. Okay, we're talking about transcription. Remember like you talked about the messenger RNA being created from the DNA? Well, in prokaryotes, RNA synthesis and protein synthesis takes place in the cytoplasm. Remember, pro means no nucleus. In eukaryotes, RNA is produced in the cell's nucleus, then moves to the cytoplasm to play a role in the production of proteins. Our focus will be on transcription in eukaryotic cells, the ones with the nucleus. Transcription has an enzyme. It requires an enzyme known as RNA polymerase that is similar to the DNA polymerase. Okay, the RNA polymerase, what it does, it unzips the DNA, remember the two strands, unzips it, and then creates the RNA, okay? So remember in DNA we had the helicase unzipped and the polymerase made the other part of the DNA, but in this case, the RNA polymerase unzips and creates. So how does this work? How does the RNA even know where to begin? Well, the RNA polymerase, remember the thing that the enzyme is going to unzip and create the RNA, binds only to promoters. So what are promoters? They're regions of DNA that have specific base sequences. So they have a code that says to the RNA polymerase, start here. Promoters are signals in the DNA molecule that show RNA polymerase exactly where to begin to make that RNA. Similar signals in DNA cause transcription to stop when a new RNA molecule is being completed. Now, here's a weird thing. RNA molecules that are being created from the DNA, okay, inside the nucleus, RNA molecules sometimes require bits and pieces to be cut out of them before they can go into action. The portion that are cut out and discarded are the introns. You think they would be called the outrons, but they're called the introns. So messenger RNA gets created more than is needed, and these introns are taken out. In eukaryotes, introns are taken out of pre-messenger RNA molecules while they're still in the nucleus. The remaining pieces are known as the exons and are spliced back together to form the final messenger RNA. Biologists don't have a complete answer to why cells use the energy to make a large RNA molecule and then throw parts of it away. Some pre-messenger RNA molecules may be cut and spliced in different ways in different tissues, making it possible for a single gene to produce several different forms of RNA. Also, this editing that goes on can also play a role later on in evolution. Okay, kids, I know this was kind of a long video, but it was very important. So as usual, have fun and keep learning.